All right, class. Today we look to get into chapter 13 today. But as we move into chapter 13, we now really start looking at the central nervous system in particular. So when we look at the central nervous system, or what we know as the CNS specifically consists of the brain and the spinal cord. Now, as we start looking at the CNS, one of the things that we look at is how the CNS develops. And that process is known as cephalization. And cephalization is the evolutionary development of the rostral, or the anterior portion of the CNS. And what that basically means is it's the evolutionary development of the brain. As we go through cephalization, we're going to see that we have an increased number of neurons that begin to develop in the brain and in the head region. And the highest level of development occurs or is reached in the human brain. So let's look at the embryonic development. We have 5 major phases, or 5 steps, that occur. We have the neural plate, which is a group of tissue, and it develops a neural groove. That neural groove deepens as the folds rise and approach one another. We have the development of the neural crest cells, and they separate from the neural folds. By the time we get to the end of the 3rd week of development, those folds meet and form the neural tube surrounding the neural canal. And that neural tube then gives rise to the brain and the spinal cord. So if we look at those phases and we look at them from a graphical representation, you can kind of see how we follow along in these 5 steps. Again, the first phase, or the first step, is that the neural plate develops the neural groove. And the cells are going to proliferate along the lateral plate margins and become the neural folds. And their tips become the neural crest cells. And so you can see this graphic here, and essentially we're going to take a slice or a cut, and we're going to open that up so that you can now look down on that. And you'll notice that when you look at this graphic, it looks relatively thick, but when you look down on it, it doesn't have a lot of depth to it. It is relatively narrow when you go front to back. But you can see that here we have the neural fold, basically right here, where we're starting to form that neural groove, and then the neural crest cells are going to arise right on the outsides of that neural groove. When we look at the second step, that gives or develops, deepens the neural groove as the folds rise and approach one another. So now we see the neural folds come out and start to wrap around, and that further develops the neural groove. In phase 3, the neural crest cells separate from the neural folds. And so here we have the neural folds developing, further developing that neural groove. And then on the side here, we see the development of the neural crest cells. As we get into step 4, and that's going to be the end of the 3rd week of development, the folds meet and they form the neural tube, and that surrounds the neural canal. So you'll notice that that neural groove is now fully engulfed. We've now formed the neural tube, and that forms this neural canal going right down the center of all of this tissue. The fusion begins in the middle and then progresses superiorly and inferiorly, so it's going both up and down. And the openings at the cranial and caudal ends of the tube close by the end of the 4th week of development. So usually around the end of the 1st month, we've already started to have good development of these particular cells and of this tissue. So as we continue to develop this embryonic tissue, we're going to see that the anterior end of the neural tube actually gives rise to the 3 primary brain vesicles. Now, these primary brain vesicles form by the late 4th week from the cranial neural tube cells. And those 3 primary brain vesicles are going to be the prosencephalon, the mesencephalon, and the rhombencephalon. Now, the prosencephalon is going to give rise to the forebrain. The mesencephalon is going to give rise to the midbrain, and the rhombencephalon gives rise to the hindbrain. And all 3 of these structures will have additional components of brain tissue that develop from those areas. Now, we have our primary brain vesicles, those three that we just talked about. But now from those three primary vesicles, we now develop five secondary brain vesicles. And these five secondary brain vesicles actually develop by about the fifth week. So it takes about a week or so for our secondary vesicles to develop. Now, if we remember, our forebrain, or the prosencephalon, is going to develop into our telencephalon and our diencephalon. Now, the telencephalon is basically going to be our cerebral hemispheres and our cerebral cortex. Our diencephalon is mostly our thalamic structures. Okay, so our thalamus, our hypothalamus, and the epithalamus. The mesencephalon is going to remain undivided, and the mesencephalon gives rise to the midbrain portion of the brainstem. The metencephalon and the myencephalon arise from the hindbrain, and they're going to give rise to the pons and the medulla oblongata, respectively, and those also are parts of the brainstem. Now, when we look at these kind of give the structures that develop from those secondary brain vesicles. We have our telencephalon that gives rise to the cerebrum, which includes the hemispheres and the cortex, the white matter, and our basal nuclei. We have the diencephalon, which, as I said earlier, gives rise to our thalamus, hypothalamus, and the epithalamus, but it also includes the retina. Our mesencephalon, again, is undivided, and it gives rise to the brainstem portion, and that's going to be the midbrain. The metencephalon gives rise to the pons, but it also includes the cerebellum. And the myencephalon gives rise to the medulla oblongata. You'll note that the midbrain, the pons, and the medulla are the three components that make up the brainstem of the brain. The central canal of the neural tube then enlarges, and it's going to form our fluid-filled ventricles that we're going to find within brain tissue. So as we go through brain development, one of the things that occurs is that we have this restriction. Obviously, the brain is developing within the skull, and there's no further areas for it to develop other than within the skull itself. And so that's where we start to see that the brain twists and folds on top of each other. And so we have what we call a midbrain flexure and a cervical flexure. And they actually cause the forebrain to move toward the brain stem. Our cerebral hemispheres, as they develop and get larger, will actually grow posteriorly and laterally. So they're growing more towards the back of the head and outward. Our cerebral hemisphere surfaces then start to crease and fold into what we know as convolutions. And as they crease and fold, it allows us to continue to develop and create more tissue in a restricted area. This graphic here shows us the development of the brain and how we see these areas over here start to fold and twist on top of each other. And so you'll notice that as it continues to develop around 13 weeks, you'll notice that things are now starting to space out. They're getting wider. They're starting to fill in the skull a little bit more. And so this is what our brain tissue, our spinal cord really start to look like really after about the third trimester. The brain then continues to develop in the fetus. You'll notice that the telencephalon envelopes the diencephalon and starts to develop these surface folds. And so we notice that at around 26 weeks here, we really are starting to get towards the end of the third trimester. And you'll start to see that we're getting these folds and these convolutions in the brain tissue itself. So as we get into the brain at birth, you'll notice that we now have more convolutions. We have these folds, we have these ridges, but now we essentially have what we are, or what we know as a regular brain itself. So here's our brain. You'll see underneath that there's some tissue that we cannot see that gets folded underneath. And then here's what we know as the brainstem, where we include this midbrain, pons, and medulla. Most of our brain tissue now have developed what we know as gyri and sulci. And we'll talk about those here coming up. But the gyri are actually the ridges of the brain, and the sulci are going to be these small little grooves that we find in the brain. And they are going to be present at birth. So after we're born and our brain continues to develop, we can start to look at the overview of what brain anatomy really looks like. And this is really looking at, for the most part, an adult brain. When we look at a brain, there are four major brain regions. We have the cerebrum, the diencephalon, the brainstem, and the cerebellum. The cerebrum is essentially our two brain hemispheres. And within the cerebrum, there are 5 lobes per hemisphere. We've already discussed that the diencephalon is mostly our thalamic structures. The brainstem we've identified as being the midbrain, the pons, and the medulla. And then the cerebellum is the structure towards the posterior aspect of the brain. When we look at the surface of brain tissue, particularly the cerebrum, gyri are going to be what we know as the ridges. So as the tissue comes up, we have the ridge, that's a gyrus. The sulci are going to be the depressions that are formed between those ridges. If they're relatively shallow, they're called sulci, or individually, they're called a sulcus. If they're deep, then we call them a fissure. So fissures are identified as a deep sulcus. Again, the anterior portion is known as the rostral side of the brain. The posterior portion is called the caudal side. So let's take a look at the human brain. This is our left lateral view. So again, there are some things that we look at. Particularly, you can see the dashed arrows or the dashed lines that help to show us where the lobes are. Now, the most common lobes are the frontal lobe that you'll see here towards the front side, the parietal lobe, which if you've gone through the anatomical bone portion, you'll notice that this falls right along the lines of the parietal bone. The occipital lobe falls right along the lines of the occipital bone, and the temporal lobe also falls along the lines of the temporal bone. Our fifth lobe is going to be one that we'll see here coming up, and it's going to be known as the insula. If we look at the other components here, you'll see that we have the brainstem, which includes the pons and the medulla. And up higher, we can't see it because it's surrounded by the other portions of the brain, would be the midbrain. Here is our cerebellum, again, more towards the inferior posterior aspect of the brain. And then all of these almost like worm-like structures that you'll see, these ridges, these are what we know as the gyri. And in between those gyri, we have those depressions that we know as sulci. Here's our inferior view, or looking up from the bottom side. Again, you get a chance to see the brainstem coming up. The portions that you couldn't see before, which include the midbrain, are more identifiable underneath. Here's your pons, which is kind of a larger section, and then the medulla oblongata as it comes down and begins to form the spinal cord. You can see a lot of the other locations where a lot of our cranial nerves are coming in. We'll talk about those later. But this is a good graphic that helps to give you an idea of where all of these structures are located. Here's our mid-sagittal view of the human brain. We've essentially taken a cut right down the center. So this is looking at one half from the interior side. And you get a chance to see how deep some of that tissue actually goes and what it looks like once you get into the deeper levels of the brain. It does show us the levels of the diencephalon, which are more difficult to see otherwise. And so you get a chance to see where we have our epithalamus, our thalamus, and the hypothalamus as we get into this section right through here. So now as we move out of the brain for just a second before we come back to it, we take a look at the organization of the CNS into the spinal cord. Now the spinal cord has this central cavity that is surrounded by a gray matter core. And the central cavity is this hole going right down the center. And it's going to be filled with cerebrospinal fluid. But around that central cavity, we have this gray matter core, or what I often refer to as the gray butterfly. This butterfly kind of gives us an idea because it's going to be very specific in where a lot of our fibers are coming in from. The external areas around that gray matter core are going to be composed of myelinated fiber tracts. And one of our words or terms that we're going to want to know is a tract. Now, specifically, the definition of a tract is going to be a collection of neuron processes found in the CNS. And this will be one of those words that I talk about that we do want to know what the definition is and make sure that we really understand it. So let's just make this our attendance term for the day, or the attendance code word, and that's going to be a tract. So now as we take a look at the brain, or we go back to the brain, it's going to have a very similar pattern with additional areas of gray matter when compared to the spinal cord. Now, nuclei are going to be found in the cerebellum and the cerebrum. And nuclei are one of those terms that we want to know. You'll notice down here I typed out all of the components, or all the 4 terms that I typically find a little bit important, and I do want you to know the definitions for. The definition of a nuclei, as we talk about the nervous system, is a collection of neuron cell bodies found in the CNS. And then we go on to the other 3. The tract, which we just identified on the previous slide, are collections of neuron processes found in the CNS. A nerve is a collection of neuron processes found in the PNS. And a ganglion are collections of neuron cell bodies found in the PNS. So please make sure that you spend a little time and really understand the definitions of those 4 terms. We also then find in the brain the cortex of the cerebellum and the cerebrum as well. So one of the other components of brain tissue are called the ventricles. Now, the ventricles don't necessarily have the brain tissue so much as they're essentially cavities within the brain. They're going to be lined with ependymal cells. And if you remember going back to our glial cells, when we talked about ependymal cells, one of the characteristics were that they were ciliated. And one of the things that we know about ventricles is that they contain CSF, or cerebrospinal fluid. One of the significance of ciliated cells then are that as the cilia move, they create this current. Remember, the purpose of cilia are to move fluids, and they do this by creating a current. And so as we have the cilia on these cells, they're actually creating a current to move cerebrospinal fluid through the ventricles. These ventricles are actually going to connect with each other and with this spinal cord central canal. And we're going to find that we have 4 ventricles, 2 lateral ventricles, a 3rd ventricle, and a 4th ventricle. So our 2 lateral ventricles are going to be the large cavities that we find within the cerebrum. Our 3rd ventricle is going to be a very narrow space in the middle of the diencephalon. Our 4th ventricle then is a sickle-shaped space that's between the pons and the cerebellum. It's connected to the 3rd ventricle by a structure known as the cerebral aqueduct. Now, the 4th ventricle opens to the subarachnoid space both medially and laterally, and we will talk about what this actually means coming up here later in this chapter. It's going to narrow before merging then with the central canal of the spinal cord. So here we take a look at the ventricles of the brain. Here are going to be our 2 lateral ventricles. Again, you see the lateral view here, the anterior view here. You'll notice how deep the lateral ventricles are as they go front to back. But you'll also notice that in this anterior view, they basically represent or appear to be almost ram horn-like. Okay, you'll see they kind of roll back and out kind of in this somewhat circular pattern. You'll notice our 3rd ventricle is relatively deep from front to back, But notice that when we get into the third ventricle, on the anterior view, it's not very wide. It's relatively narrow. But then the reverse is true for the fourth ventricle. Notice the fourth ventricle is a little bit wider through here, but then also notice where it is here, and it's not very deep from front to back. And then we have this connection then going all the way down into the central canal of the spinal cord. And then here you'll see from the third ventricle to the fourth ventricle, we have a smaller passageway, if you will, and that's going to be our cerebral aqueduct. So let's look at some of the areas of the brain. And we're going to talk about the largest one first. This is going to be the cerebrum. The cerebrum is the origin of all of our complex intellectual functions. We have two large hemispheres on the superior aspect of the brain. And it's going to be the center of intelligence and reasoning, thought, memory, and judgment. And it's also going to be the location of our voluntary motor, visual, and auditory activities. As we look further into the cerebrum, we look at some of those surface markings. Now, we've talked about the ridges, which are known as gyri, and we talked about the shallow grooves, which we know as sulci. We also did talk about fissures, which are the deep grooves. And then we touched on the 5 lobes: the frontal, the parietal, the temporal, the occipital, and the insula. So some of our specific surface markings are going to involve our sulci, our fissures, and our gyri. We look at the central sulcus first. And the central sulcus is going to separate the precentral gyrus of the frontal lobe and the postcentral gyrus of the parietal lobe. So this basically is the dividing line of the 2 lobes. But make sure we understand the terminology. The precentral gyrus is going to be the gyrus that's in front of the central sulcus. The postcentral gyrus is going to be the gyrus that's after the central sulcus. Remember, a gyrus is essentially one of those ridges. The longitudinal fissure is going to be this deep cavity or this deep crease that helps us separate the 2 hemispheres. So it gives rise to the right and the left hemispheres of the brain. We also have a transverse cerebral fissure, and that separates the cerebrum and the cerebellum. So if we take a look at the superior aspect of the brain, here we have our frontal lobe, kind of in this tan color. Here we have our parietal lobe in this pink color. You'll notice right here is our central sulcus. Again, that is dividing the frontal and the parietal lobes. Here, this ridge is our precentral gyrus. So we have the ridge just in front of, or just anterior to the central sulcus. On the back side here, we have our postcentral gyrus. Again, that's going to be posteriorly then to the central sulcus. Dividing the right side from the left is going to be that longitudinal fissure. Again, we have our 5 lobes, 4 named for the overlying cranial bones: the frontal, parietal, temporal, and occipital. And the insular lobe is not visible at the surface because we basically have to pull it back in order to see it. The frontal lobe is the anterior part of the cerebrum. It gives rise or has or contains the lateral sulcus, and it separates the inferior frontal lobe from the temporal lobe. The frontal lobe has varied functions. It's going to include motor control. It has function for concentration, verbal communication, decision making, planning, as well as personality. Our parietal lobe makes up the superior posterior part of the cerebrum. The anterior border of the parietal lobe is the central sulcus, and that contains the postcentral gyrus, and it's that ridge that's just posterior to the central sulcus. The parietal lobe serves as the general sensory function area. For example, trying to evaluate different shapes and textures of different objects. The parietal lobe is internal to the temporal bone. It's going to be located inferior to the lateral sulcus, and it's going to function in hearing and your sense of smell. The occipital lobe is the posterior part of the cerebrum, it's going to function in vision and visual memories. And then, as we said, the insula is that fifth lobe. It's going to be deep to the lateral sulcus, and it's a small lobe that can actually be observed by pulling away the temporal lobe and the surface of the cerebrum. And it's going to function in memory as well as the sense of taste. So as we get into the cortex, we get into a process known as lateralization of cortical function. And what that means is that each hemisphere is going to be a little bit more controlled for certain activities. So for example, the left hemisphere typically controls language, math, and logic. The right hemisphere tends to result in insight functions or spatial visual skills, intuition, and artistic skills. However, they do communicate with each other. So the left and right hemispheres communicate via fiber tracts in the cerebral white matter. And again, you'll see that word there again, tracts. So these are going to be collections of processes that we find within the CNS. So let's look at the structure a little bit more of the cerebral cortex. It's going to be a thin, superficial layer of gray matter that's really about 2 to 4 millimeters thick. It's going to be about 40% of the mass of the brain, and it's going to be the site of conscious mind. So things like awareness, sensory perception, voluntary motor initiation, communication, memory storage, and understanding. Again, these are all things that are involved with consciousness, things that you do while you're awake and while you're aware of what's going on around you. Each hemisphere, both the right and the left side, is going to connect to the contralateral side of the body. And so what that means is, if you've ever heard the phrase, your right side controls your left side and your left side controls your right side, there's a lot of truth to that, because when it comes to motor control, specifically, your right side actually does control what your left side does, and vice versa. Your left side controls what your right side does. And so, we've talked about lateralization. And so there is lateralization of cortical function in the hemispheres. So when we look at the functional areas of the cerebral cortex, there are 3 types of functional areas. We have motor areas, sensory areas, and association areas. Our motor areas are going to control voluntary movement, things that you have control of from a conscious perspective. Sensory areas are the body's conscious awareness of sensation. It means you understand and you can feel what's going on around you. And then we have our association areas. And the association areas help to integrate diverse information. They also help to store memories and associate those memories with certain other sensations. The cerebral cortex also is conscious behavior, and this conscious behavior involves the entire cortex working together. And all of these areas are made of interneurons. And if you remember, interneurons are a functional class of neuron, and they make up 99% of our functional neurons, from motor neurons, which is one type, sensory neurons, another type, and then the interneurons, which are the association components. So let's look at a few specific motor areas. While there are numerous areas, we're actually going to talk about 4 of them. We're going to talk about the primary somatic motor cortex, the premotor cortex, Broca's eye field, and the frontal eye field. Now, the motor areas are going to be housed within the frontal lobes. Our primary motor cortex is specifically located in the precentral gyrus. It's also called the somatic motor area. And if you remember, one of the things we talked about with somatic is you should think skin, skeletal muscle, and joint. So it's going to control the skeletal muscle activity on the opposite side of the body. And what it does is it projects contralateral fibers to the opposite side within the brainstem or the spinal cord. So when we look at this term contralateral, contra means opposite. So we are projecting fibers towards that opposite side. The controlled body regions map then looks as if we call it a motor homunculus, which a definition of a motor homunculus is kind of an upside down caricature that represents the motor innervation of body regions. So here's our primary motor cortex. Again, specifically in the area, we are going to want to know where this is. It falls again within that precentral gyrus. Notice where we are in the brain. Here's our anterior side. Here's our posterior side. Here's our central sulcus. And this then is our precentral gyrus. You'll notice that we're now taking a coronal cut where this area where the foot looks like it is actually going down right into the longitudinal fissure. This area then is the lateral aspect of the brain. So you'll notice this kind of caricature. You'll notice the components of the body that look the largest, and those make up the largest area of the brain. You'll notice the hand has a lot of fibers to it. It makes up a pretty big section because we have a lot of fine muscle control, and it's very detailed. Same thing in the face, very specific movements, lots of connections, lots of control. And so this is what the areas that are controlled by the brain, where they're located. And so you can kind of take some moments to look that over. You don't have to memorize it, but just get an idea of where some of these structures really are. So as we continue on with the motor areas, we start to look at the motor speech area, which we know as Broca's area. It's going to be located in the inferior lateral portion of the left frontal lobe in most people. And it controls motor movements for vocalization. Our frontal eye field is on the superior surface of the middle frontal gyrus. It regulates eye movements needed for reading and binocular vision. And then our last functional area is the premotor cortex, or what we also know as the somatic motor association area. It's going to be located anterior to the primary motor cortex, and it controls coordinated learned and skilled activities. So again, please note the location is anterior to the primary motor cortex, which is located in that precentral gyrus, and also note what its function is. It's a somatic motor association area, which means it's involved with the movements of skin, skeletal muscle, and joints. But what it's really doing is it's an association area. And so it utilizes previous memories to help coordinate these learned and these skilled activities. So then we move from our motor areas to our sensory areas. So now we're looking at the area known as the primary somatosensory cortex. This is our first one. And it's going to be located in the post-central gyrus of the parietal lobes. It's going to receive somatic sensory information from essentially the rest of our body, or from outside of our body. So we're utilizing receptors that are in our skin, that we find in our muscles, that we find in our tendons, our ligaments. And so we're really looking at things like proprioception that help the brain understand where we are in space. It takes information from touch, from pressure, from pain, and from temperature receptors. And it gives us what we know as a sensory homunculi, very similar to our motor homunculi. The somatosensory association area then is an association area that's closely linked to the primary somatosensory cortex. It's going to be immediately posterior to the post-central gyrus in the parietal lobe. And it's going to integrate the touch information, allowing us to identify objects by feel. And it determines size, texture, and relationship of the parts of objects being felt. So now I'm going to go back to this, and we're really looking at it somewhat of an easy way to understand this. When we look at these primary sensory areas, for example, this one is the primary somatosensory cortex. It basically is receiving sensory information from the body. The association areas are the areas that really help to store memories. And it's because of these association areas that allow us to identify, in this particular case, what we're actually feeling. So if I pick up, let's say, for example, my cell phone and I have my eyes closed, I'm assuming most of you could do this. You pick up your cell phone, you pick up a remote control, you pick up a pen with your eyes closed, you hold it in your hands for a second and move it around. The sensation you get from holding that gives you a lot of information, from the shape to the structure to the size to the texture, and it allows you to identify what that object is without actually seeing the object, because you held a pen or a pencil or a remote control long enough or enough times in your life to have those memories stored. And so your brain then processes what it feels from a sensation perspective and compares that to memories that it has about those particular characteristics. And that's how you're able to identify those particular objects. So here's our sensory homunculi. Again, we get the same thing. Here's our brain. Here's the anterior portion, our posterior portion. Here's our central sulcus. Here's our post-central gyrus there in red. This right through here indicates our longitudinal fissure. And you'll notice that again through the homunculi, some of these areas that have larger areas or coverage areas on the homunculi. Your face, because it's very sensitive. Your hands, because we need to be very particular about sensation coming in from our hands. You'll notice those take up some of the biggest areas in this homunculi. Some of our other sensory functional areas. Again, you'll notice that a lot of these have the primary cortex and then an association area with it. So when we look at the primary visual cortex, it's going to be located within the occipital lobe. The visual association area surrounds that primary visual cortex, and it integrates color, form, and these memories to allow us to identify things that we see. For example, faces are a perfect example. You get a chance to see somebody's face, you get a chance to see their eyes, their nose, their mouth, their face structure. All of that put together have formed memories that allow us to compare that to the information that's actually coming into the brain. Same thing with our auditory areas. We have a primary auditory cortex that's located within the temporal lobe. We also have our auditory association area. It is also located in the temporal lobe, and it interprets sounds. And then it also stores and retrieves the memories of sounds. One of the best examples I can give of this is when you talk on the phone. You know, when somebody calls and you pick it up, you know, imagine mom or dad or grandma or grandpa making a phone call to you and you answer it. Very often you know right away without looking at caller ID nowadays, but you'll know right away most of the time who that is based on the sound of their voice. You can't see them, but you can hear the pitch. You can hear how fast they speak. You can hear all of those characteristics of voice and audio, and that helps you to process the information and identify that who that particular individual is. We also have the primary olfactory cortex, which is located within the temporal lobe. It provides us with conscious awareness of smells. The primary gustatory cortex is located within the insula, and it's involved in the processing of taste information. Some of our other functional brain regions that help us to integrate all the information from a lot of our multiple association areas. One of these areas is the prefrontal cortex, and it's located rostral to the premotor cortex in the frontal lobe. And it's involved with complex thought, judgment, personality, planning, and decision making. And it's still developing in adolescence. Wernicke's area is typically located in the left hemisphere, and it's involved in language comprehension. And this is where we'll actually stop and pick up the next time. We'll try to get chapter 13 finished. If not, we'll still have a little bit of chapter 13 to finish in the following week.